tissue here. So what organ do you think this is, looking at it at scanning view? What do you think? I heard the answer. That is the spinal cord. Okay? So when we talk about the histology of the nervous system, usually the nervous system is divided into two types of matter. We have the gray matter and the white matter. Okay? It's quite obvious when you look at it grossly, the gray, but unfortunately in histology, everything is pink. We don't see gray and white. So what's the difference between gray matter and white matter in terms of histology? What do you think? Is this here gray or white matter? And how about here? Okay, that's correct. So in the spinal cord, the central portion is forming this H-shaped like structure, or sometimes authors call it like a butterfly. It's like a butterfly or letter H. This central portion is the gray matter, and the peripheral portions are the white matter. Okay? In terms of histology, it's gray because it's going to have the nerve cell bodies. Here are the neurons, these large cells here. These are the neurons. Whereas in the white, it's whiter because most of the tissues here are actually going to be axons surrounded by their myelin sheath. And myelin sheaths are lipids, so they're lighter in color and appearance. Okay? So this is the gray matter, this is the white matter. Okay? Now, each of these areas of the gray matter form like a projection. So there's a projection here, and a projection up here, and then this one's a little chopped off, but imagine that it's here. So these projections are called the horns. Okay, they look like horns. We have the anterior horns here, and then we have the posterior horns here. The anterior horns have larger neurons here, okay? And these neurons are motor neurons, okay? Whereas the ones in the posterior are sensory and they're smaller. Okay? So let's take a look at some of the cells here. So here's the low power, and then we go to scanning, high power. So these are the neurons. The staining is a little bit faded, not so good. Okay. You'll notice that the neurons, or the cell body of the neurons, are here, and they have several processes. Okay. The processes that receive information and send them to the cell body are called what is it called? That receive and send information to the cell body, dendrites. And then the one that takes the information and away, axons. So the way to tell the difference in terms of histology is axons. If you notice the cell body, they have this dark sandy-like appearance that's granular, dark blue here. It's because there are a lot of ribosomes. And ribosomes really stain well with hematoxylin. So giving a nice blue granular appearance. Okay. But the axon doesn't have these little blue granules going into it. That's called the axon hilum. So this here is the axon. Here is the axon. Dendrites, on the other hand, they still have this dark granular appearance. Okay. So that's the axon. Here's another axon. Dendrite. Now for the white matter, the staining is really bad, so it's kind of hard to really appreciate the white matter. Okay. What you see are actually cross-sections of axons. So there's an axon here, this little dark circle, surrounded by a lighter pale myelin sheath. Okay. That's mostly what the white matter is going to be here, as well as their supporting cells. We'll look at the new slide, because the new slide has a little bit better staining. Okay. So here's the new slide. The staining is very nice here. Okay. So here, what type of matter is here? Gray. Here, white. Okay. This one actually still has portions of the meninges. So what are the layers, the parts of the meninges? This one that's really thick and prominent and composed of dense, irregular, collagenous connective tissue is the dura matter. So dura means tough. Matter means mother. So this is the tough protective mother that's protecting everything. And then you have this one here that's a little bit spacious and has these little thread-like things, reminiscent of a spider web. That's your arachnoid. And then on the surface, directly on top, you have this very thin layer. That would be your ileum mater, right there. Little very thin layer. Okay? In which layer would you find your cerebrospinal fluid? Subarachnoid. See the spaces here, underneath the arachnoid, subarachnoid. Okay? Alright, so let's look again at the cells. So we're looking at gray matter. How can you tell it's gray matter? Because of the presence of neurons. Okay. Now, if you look at the background, there's a lot of these pink lines, right? All these pink lines going in many different directions, right? All this pink that you see is called neurophil. Okay? It's called neurophil. What neurophil is most of the cells of the nervous system, they have processes, okay? They have, like, the neurons, they have axons, dendrites, even the oligodendrocytes and the astrocytes, they all have processes. And these processes are intermingling with one another, forming all these little lines, these pink lines that you see. So that's the neurophil. Okay? It's just a bunch of all the processes from all of the supporting and the neurons. Okay? So that's neurophil. So here, looking at a neuron. So let's look at some of the supporting cells. Okay? So aside from the neurons, you have supporting cells which are called glial cells. And there's actually a lot of different glial cells. And I will require you to be able to identify them and know their function. Okay? So the most common of the glial cells you'll see are these. Well, let's not start with that one. Let's do the easiest one to identify. Okay? The easiest one to identify here is this one. It has the round nucleus, but it has this nice clear rim of cytoplasm. Okay? So it looks like what we call as the fried egg appearance, or it looks like a sunny side up egg. Okay? Here's another one. See the white cytoplasm? What do you think these cells are? And why do they have that white cytoplasm here? What do you think? Here's another one. What is this fried egg cell looking? That's your oligodendrocytes. Now, next question. Why does it have a pale cytoplasm? What's the function of oligodendrocytes? Yes. They make the myelin sheath in the central nervous system. And myelins are lipids. Again, staining very poorly with H&E. So that's the fried egg cell that you see here, oligodendrocytes. What makes the myelin sheath in the peripheral nervous system? Schwann cell. They're a little different because oligodendrocytes have processes, and they can wrap through several axons. They can wrap around several different axons, and even on the same axon several times. Whereas in Schwann cells, only one Schwann cell could only cover a small portion of the axon at a time. So you'll need several Schwann cells to cover a great amount of the axon there. Okay? So this is the easiest one to identify with the fried egg appearance. That's the oligo. Now the other cells here, you notice, they don't have the white cytoplasm. See, like this one, and actually, to be honest, they're actually more numerous than your oligo. What are these cells here? Here. What are they? Astrocytes. Good. Astrocytes have a lot of different functions, okay? The main role is they also have processes too. But when they have the processes, they'll wrap around other cells. And they'll even wrap around blood vessels. So their main function, you can actually see there's a little capillary here. There's this astrocyte here, wrapping around it. They actually have the processes that wrap around it, okay? And their main role is for structural support, for structure. They hold things in place. But they also have other functions. Because they wrap around the capillaries, they're important in forming the blood-brain barrier. They're one of the components of the blood-brain barrier, together with the lining endothelium and the basement membrane. And the astrocytes. Those are that's your blood-brain barrier. Okay? Now, the other cell, which is really hard to find, which I don't think I want to struggle to look for it, is the microglia. Okay? Now, microglia is actually a type It's an immune cell, and it does phagocytosis. It's a little different from your astrocytes because the nucleus is very slender. But again, they're a little bit harder to see, so I'm not going to require you to find it because you may struggle to find it. Okay? Although I did find it in my other section, but it may take a while. Okay? So just know the function of the microglia later. We have another cell that you should know that's also seen in the spinal cord. But let's look at the special structure in the spinal cord. Okay? This structure here. What is this structure called? That is the central canal. Okay? So the central canal will also have CSF inside. So this is the CSF. And then if we look at it, it looks like it has a lining epithelium that's even like columnar or cuboidal in shape, right? Sometimes they even have microvilli. But these are not epithelium. What are these cells? Ependymal cells. They're not epithelium because there's no basement membrane. Okay? The epithelia are supposed to always rest on a basement membrane. But here, it's immediately in contact with the gray matter here. So it's immediately in contact with the gray matter. So that's the ependymal cells. Ependymal cells will line the central canal, the ventricles, as well as the choroid plexus. Okay? Now let's look at the white matter. Okay? So here's the white matter. Okay? And I know it's a little bit confusing, but this is an axon. This is a cross section of an axon. And then there's a myelin sheet surrounding it. Okay? So these are axons that are going up and down the spinal cord. Axon and then the little myelin sheet. And then the supporting cells are usually astrocytes. Okay? Usually astrocytes. And oligodendrocytes. Oligodendrocytes. Since this is the spinal cord. So it's oligodendrocytes and astrocytes. Okay? Do you see neurons here? There's no neurons. Okay. So that's the difference. Unlike here, you already see the neurons. Okay, with the plastic fish eye nucleus. It's better in the motor because they're larger. Let's go to the motor area. See the nerve cells here? Much larger. Here's the classic fish eye, pale vesicular nucleus, and a single prominent nucleolus. Fish eye. Okay. And you can see the somewhat granular blue cytoplasm due to the distal bodies. Axon hillock is here, so this is the dendrite. This is the paler one. Okay. Alright, so that is going, we have something. This one, see that flattened cell? It's not a squamous cell. That's a microglia. The nucleus are more flattened and elongated. So that one is your immune cell. Okay? They're a little challenging to find. So we found one. Okay? Alright. So that is the spinal cord. So technically the spinal cord has different histologies based on what you're looking at. Cervical, thoracic, lumbar. And there's also all those tracts. But I'm going to leave that to Dr. Klein to teach you that. I just want you to know the generals. Okay? Know the white matter, the gray matter, the cells, and the general arrangements. Okay? Alright, so that's the spinal cord. Let's move on to the next structure we have. Okay. Now we have something here. I'll talk right now. Hold on. Okay. We've got a very intimidating structure here. It's very complicated. Did you notice that it's a little different here from here? So this one is different from the tissue here and also from here. Okay. So in the spinal cord, what's the arrangement of the gray matter? The central portion is the gray and the peripheral are the white matter, right? But here we have a different structure and its arrangement is different. So let's go to the surface. Here's the dura mater. See the dura mater? And see the little spider web-like thingies? This is the arachnoid. And then on the surface would be the pia. So this is the meninges. Dura, arachnoid, pia. Okay? And then we have this. And then the deeper portions, we have this. So this portion here, if we were to go into a little, zoom in a little bit. See again, we see the fisheye nucleus. So these are neurons. Although they're not as big as the one that you saw in the spinal cord, because those are really motor neurons, they're big. But these are neurons here, although they're a bit smaller. And again, you can see also the supporting cells. Okay? Let's see if we can find some supporting cells. There's that fried egg appearance. So this is the oligo. Then there's one that doesn't have that white cytoplasm, that's the astro. Okay? And then the ones, these are small neurons with the fish eye nucleus. Okay? So what type of matter am I showing you here? This is gray matter. Although the neurons are smaller, this is gray matter. Okay? And then the background is neurophil. Okay? And then let's go to the deeper portion. So this outer portion is backwards. It's different from the spinal cord because what was on the surface of the spinal cord? It's white matter. But here, what's on the surface? Gray matter. And if we go deeper, what's the middle portion? Let's look at it here. Let's zoom in. So now we are at high power. Most of these are astrocytes. Okay? And you see the fibers going this way? These are usually axons again. So this is going to be white matter. This is white matter. Okay? Again, there's an oligo, astro, okay? But there's no neurons. You don't see the neurons. Okay? So this organ that we're looking at, what do you think is this organ? What do you think? That's here, has this really thick gray matter, and then the middle portion is white matter. What's your guess? So this is the cerebrum. Okay? This is the cerebrum, the main bulk of your brain. Now, if I want to be really cruel, there are actually several layers of the cerebrum. There's a layer here, there's another layer here, another layer here, but I'm not going to ask you to memorize those layers. Okay? Maybe Dr. Klein will ask you the layers, but I will not ask you the layers. All I want you to do is differentiate gray matter from white matter and identify the cells and know that this is a cerebrum. I don't really mind if you don't know this is the molecular, this is the outer Purkinje, or all those 11, or outer pyramidal layer. It's okay. Let's look around again. Oh, see something. It's not very difficult. You'll notice that sometimes you'll see, let's see if we can find it. Here. Okay. All right. Sometimes you see these large neurons that are kind of shaped like a triangle. That's a pyramidal neuron. They have a prominent, they're triangular shape. And they usually will all point in the same direction. Let's see if we can find another one. Pyramidal neurons. They're major excitatory neurons. You can find them. But I'm not really going to ask you to identify them. But they're just bonus. Alright, so that's your cerebrum. Alright. I will ask you though to really know this structure because it's easier. This one's easier. Oh, maybe I should show you the old slide first. Here's the old slide. Then I'll show you the new slide. So the middle portion here is, let's look at this middle portion, the very core. really pink color. Do you see neurons in here? No. You just see fibers going in the same direction. So what type of matter is this? White matter. So this organ also has the same arrangement of the gray and white matter. The central portion is white, the peripheral portion is gray matter. Okay? And you'll notice it's kind of rolled into these folds here. Gyran, sultan. So this organ is what organ is this? This is the cerebellum. Cerebellum. Okay? Now, the gray matter, it also has layers, but there's only 3. So I'm going to require you to memorize it. There's only 3 layers. Okay? It's not as bad as the cerebrum. Okay? The cerebrum has a lot of layers. Cerebellum is easier because the gray matter only has 3 layers. So let's look at the layers. Here's part of the dura matter here. Okay? So this first layer of the gray matter, the outermost layer, is from here to here. What is this layer called? Anyone? This outermost layer of this. And this outermost layer is going to be mostly neurophil with very small neurons. These are very small neurons. So this is called the, what's your answer? Don't know? Oh, such a shame. Okay, why? Hey, this is your molecular layer. Okay, it's your molecular layer. Next layer, we see some very large cells here and here. And they actually have dendrites that extend. In fact, the dendrites of these cells are actually what's forming most of the neurophil here. So, see, look at the branches of these dendrites of these cells. What is this cell called here? What are these cells? Anyone? Very large cells. And they're the main cells of this cerebellum. This one here. So, what's the answer? Yes, I heard it. It's Purkinje cells. They are the main cells of the cerebellum. This is our Purkinje cell. So therefore, what's the name of this layer? Purkinje cell layer. Easy, Purkinje cell layer. And then if we go here, it looks like they're little granules, right? Looks like tiny little granules, but these are not granules. These are very small neurons. Okay, very tiny neurons, giving it a granular appearance. So what do you think is the name of this layer? It's the granular layer, or the granular cell layer. And then the innermost layer is here, the white matter. Okay? So that, not there. It's actually here. Here. The white matter. Okay? So let's review the layers now that I told you the answer. This layer is molecular, and then this ones that have this cell, what is it? And then this one, granular, and then the middle, white matter. Let's compare that to the new slide. Very clear, better staining. See what type of matter is this? White matter. So again, white matter, will it have the nerve cell bodies? No. Instead, they're mostly axons and the supporting cells. Let's take a look again. Very nice. So we see a lot of these fried egg cells that are oligo, and then the rest are astro. And then if you're lucky, might see a microglia, possibly, no, not really. Usually thinner and longer. Yeah, don't struggle for that. So let's go back to the layers in the new slide. So looking at the new slide, we have, okay, let's look here. We'll go to higher power. Alright, so what is this layer here? Molecular layer. So it's mostly neurophil and a few scattered small neurons and supporting cells. And then take a look, there's that fish-eye nucleus of the nerve cells. So these are Purkinje cell, that's your Purkinje cell layer. And then this one, you see with all the small nuclei of these small neurons, this is your granular layer. And finally, the other. Okay? Alright, so that's the cerebellum. Next. This one. Let's do the other one first. Let's do this one. Okay, so what do you think we're looking at here? What do you think? You see a lot of these circular structures. What are these circular structures? So these are nerve fascicles. Nerve fascicles. Okay. So what do you think we're looking at now that we see a lot of these nerve fascicles? Fascicle. This is a cross section of a peripheral nerve. It's hard to tell which nerve because they all histologically look the same. So sciatic, radial nerve, all of those nerves, whatever nerves you want to call them. But they're histologically the same. So a nerve, like those yellow lines that you see on the anatomy drawing, those are the nerves. So a nerve are composed of several nerve fascicles here. These nerve fascicles will contain axons of nerve cells that are going up and down the nervous system. Okay, up and down the nervous system. So let's look at the arrangement of this nerve, this peripheral nerve. Okay? Because there are some connective tissue that you need to know. The entire nerve, peripheral nerve, or the entire nerve itself, is going to be surrounded by this dense, irregular, collagenous connective tissue. You see here? This is called the epineuron. Okay? It's the epineuron. And then if we look at the individual nerve fascicle, the nerve fascicle itself also has another connective tissue. See this dark pink line? What is this connective tissue called? Perineuron. And then, I know the staining is bad, but we really can't see the individual axons because the staining is quite bad. This would be an axon with its myelin sheath, and then the whole axon will also be covered by another connective tissue, which is the endomeuron. Okay? And then sometimes we see some cells here, the nuclei of these cells. These cells are making the myelin sheath of the peripheral nerve. What are those cells? Schwann cells. Okay? So that's a cross section of a peripheral nerve. We also have another section. Let's look at this one. Is it this one or the other side? Awesome. This one is a newer slide. We also have a small nerve here. So see the nerve fascicles? But this one is better stained because now we can really see the axon in these nerves. All right, take a look at this. You see this little dot? See that dot right there? This is the axon. This is the cross section of the axon. And you notice it has this white area around it? That's the myelin sheath. It's lipid. So, and then you see this little nucleus of the cell? That's the Schwann cell. So Schwann cell, axon, myelin sheath. And then the whole thing here will be surrounded by the endomeuron. So again, axon, myelin sheath, endomeuron. And then we have the nuclei of the Schwann cells. Axon, myelin sheath, nucleus of Schwann cell, surrounding it, endomeuron. Okay? And then, what is this connective tissue here then? This is the perineuron, because it's surrounding the nerve fascicle. And then here on the side, this is the epi. Okay? So this is another cross section of the axon. Very beautiful. You can actually see the axon, cross of the axon here. See? Axon, myelin sheath. There's the endomeuron. You see it? Endomeuron, the little thin line. Okay? That's the thin line is the endomeuron. And we see some Schwann cells. Most of these are Schwann cells. Okay? We also have a longitudinal view of a single nerve vesicle. So here's a longitudinal view. Before you saw a cross-section, now we're looking at a longitudinal. So here, what is this connective tissue? This is the perineuron. So if you look at the cross-section of a nerve, you'll see that the axons are going this way, up and down in this case. They usually have a wavy-like appearance. Okay? Let's see a very good, if we can find a very good, a perfectly cut one. Because sometimes the cut is not exactly perfect. Okay. Here, take a look. See this thin black line or this thin red line? This is a longitudinal section of the axon. And you notice it's pale on the sides because of the myelin sheath. Okay? So again, that's a longitudinal section. And then these nuclei are again usually Schwann cells. Although there may be some astrocytes, but most of them are Schwann cells. Okay? Now, if you're lucky, let's see if we can find one, and if we're lucky, you might see a portion where the myelin sheath will pinch. And where it pinches, that's going to be your node of Ranvier. Okay? But this is probably not the best slide to study because it's kind of not a very good one. So I don't want to struggle to look for it. Node of Ranvier. And I'm not going to struggle for it. Okay, but let's go over here. We see these round structures. Let's take a look at them. What are these things exactly? Here they are. And let's go to high power. There's the fisheye nucleus. See? There's the fisheye nucleus. So this is another neuron. This isn't any ordinary neuron. What are these neurons called? Do you know? And what exactly are we looking at? What is this whole structure that we're looking at? What's your idea? What are we looking at? Anyone? Yes, I heard the answer. This is a ganglion. So what exactly is a ganglion? A ganglion is a collection of nerve cell bodies in the peripheral nervous system. You can usually find them next to the spinal cord, forming the sympathetic ganglion chain, okay, for example. And what do you call these cells? These cells would be, of course, now the ganglion cells. And then if you look carefully, the cells, these smaller cells at the side, they're actually a very special type of astrocytes, but we call them as satellite cells because they're kind of going around like a satellite and orbiting the ganglion cells. And they're there to support the ganglion cells, provide nourishment and regulate the environment. Then some of these ganglion cells, like this one, it has this brown pigment in the cytoplasm. Do you know what's the name of that pigment? Lipofuscin, that's your wear and tear pigment. Okay? No function, just a sign of cellular aging. Wear and tear pigment. So that's a ganglion cell. But what do you call a collection of nerve cell bodies in the central nervous system? If it's in the peripheral, it's called a ganglia. If it's in the central nervous system, like in the brain, what do you call that collection of nerve cell bodies? It is called a... It's called a nucleus. Okay? Have you ever heard of the red nucleus or the other nucleus? Those are collections of nerve cell nerve cell bodies within the central nervous system. Okay? There's the axon, myelin sheath. Okay? That's the old slide. Let's compare that to the new slide. Here's the new slide. And you see the nice ganglion cells? Even more prominent in missile bodies. More prominent. Very nice fisheye nucleus, single prominent nucleolus, pale vesicular cytoplasm. Ganglion cells, and these are the satellite cells. Okay? Very nice. Fisheye nucleus. Alright. So when we talk about neurons, neurons are classified, they can be classified based on their morphology. For example, there's like multipolar neuron, bipolar neuron, etc., etc. What's the morphology of a ganglion cell? What type of neuron is a ganglion cell? Do you know? What type of neuron is a ganglion cell? Based on morphology. Anyone? It's pseudo-unipolar, meaning they have one process which will then actually branch into two. Okay, so that's the pseudo-unipolar neuron. And then here we have a longitudinal section of a peripheral nerve. Okay? So you can see the axon and the myelin sheath. Okay? Axon and myelin sheath. Very nice. All right. So that's the ganglion. We also have another thing I want to show you. We have another, I forgot to show you this one earlier, but this is another spinal cord. But this is not stained with H&E. This is blue. So what picks up this dye are actually the neurons. So this is showing you the cell processes of the neurons. You can't see the other supporting cells because it's usually the neurons that take up the cell. Okay? Axon, dendrites. Okay? This is again part of your knee slide. And these are smaller nerves on the background. Okay? Alright, so this is another spinal cord. Pretty, right? Yeah, histology is very beautiful. Alright, and then we have another thing that I wanted to show you. Let's see how good you truly are. What organ am I showing you? What do you see? Do you see goblet cells? So what organ is it? Intestines is not good enough. You have to be specific. Do you see villi? Are there villi, finger-like projections? No. This is not the small intestines. This is the large intestines. Why am I showing you the large intestines? Because there's also the enteric nervous system. Oh. Okay. So within the digestive system, there are two plexuses. Do you remember those plexuses that we mentioned a while ago? There's the way back in the digestive. Although the staining here is very bad, you can barely see anything. Let's go through the layers. This is the mucosa, submucosa, muscularis propria. Okay? So the easier one to find is in the muscularis propria, in between the layers, the inner longitudinal arm, the inner circular outer longitudinal. Alright. And it's really very pale staining. Let's see what we can do with it. Yeah, it's very pale staining. But this is the Auerbach plexus, or the myenteric plexus. Maybe the new slide will be better, because it's a little bit, I think it's very hard to appreciate. So let's look at it here with the new slide. Let's look at it in the new slide. Yeah, very nice. Crypts of Libericum. Crypts of Libericum, intestinal glands. Here's the mucosa, muscularis mucosae, submucosa, muscularis. So you see that there are two layers. Here's one layer, here's the other layer. You see these white structures in between? That's your Auerbach plexus. And here it is, Auerbach plexus. Now there's something you need to find in the Auerbach plexus. This cell. Auerbach plexus or the myenteric plexus. And this cell. See the cell? Again, it has that fisheye nucleus. What do you think this cell is? It's also a ganglion cell. Another pseudo-unipolar cell. It's the same. It's a ganglion cell. So what's the function of this Auerbach plexus? Here, the whole thing. What's its function? What do you think they do? Are they for decoration? Think, it's in the muscular layer. So what do you think? Why would it be in the muscular layer? Because these neurons is responsible for initiating propulsion, peristalsis. Okay? They're kind of like the pacemakers of the GI tract. Okay? They're the pacemakers. Now, it's very important. You have a very interesting condition. Let's correlate this clinically. So let's go back to the Auerbach plexus that we saw earlier. See the ganglion cell? So there is a condition that you can sometimes see in newborns where these ganglion cells fail to develop. Okay? And because there are no ganglion cells, the large intestines will not undergo peristalsis. So the baby will have no bowel movement. And because there's no bowel movement, what's going to happen to the abdomen? It's going to expand, enlarge, dilate. And if left untreated, it creates a very large colon. And that's what we call aganglionic megacolon, also known as Hirschsprung's disease. Okay? When a pathologist needs a specimen, we need to be able to identify these ganglion cells. Okay? If they're absent, then that means the surgeon has to go higher, make the resection higher. Okay? That's the clinical correlation to that, Hirschsprung's disease. Okay? Now, there's another plexus that's actually found in the submucosa, but it's not that easy to find because it has to be cut perfectly. Okay? And what's the name of the plexus found in the submucosa? Submucosal plexus, if you don't know how to spell the other term, but what's the other term? It's Meissner's plexus. Not to be confused with your Meissner's corpuscle. Where do you find Meissner's corpuscle? It's in the skin. So it's different, the Meissner's plexus and the Meissner's corpuscle, because they were discovered by the same person. So he named it both after himself. Okay? So if you discover things, name it after yourself so you'll be famous. Okay? So unfortunately, this slide doesn't have it, so we'll just have to imagine. But the structure is still the same. Look for the ganglion cell. The only difference is it's in the submucosa. Okay? And the function of the Meissner's plexus is to innervate the glands as well as the muscularis mucosae. Okay? All right. So those are the parts of the nervous system. Are there any questions